Good afternoon, everybody. We're going to get started. It's right now at 1 o'clock. My name is Abby Bronson, and I'm the SVP of Research Strategy at Parent Project Muscular Dystrophy. I'm delighted to be able to introduce this afternoon's webinar on gene therapy for Duchenne. With gene therapy having some recent successes in some other diseases, and I think you'll hear a little bit more about that in the second portion of the webinar when Dr. Sweeney speaks. With those successes and some advances made in AAV delivery, we know that there's a lot of momentum and interest in this field and in Duchenne specifically. So over the next few months, we're going to be hosting a series of webinars which are intended to help you understand what gene therapy is and, as the different trials get closer to the clinic, to be able to better understand each program. And our intent and our goal is to help you as families make better choices for everyone in your family. So the first webinar of this series and on today's webinar will focus on ensuring that everyone has a basic understanding of how gene therapy works, who it's intended for, and taking a deep dive into a few of the concepts that might be confusing, like what's the difference between micro-dystrophin and mini-dystrophin, some definitions for promoter, cassette work you've heard, and why might some trials exclude some patients, because we've also heard that gene therapy is for everybody. So we're going to try to dive into those questions. This webinar is intended to be purely educational, so going forward when specific trials are being discussed, you'll be able to better understand them. So that's our goal for today. A few housekeeping items before we move on. We're going to have time, hopefully, for questions at the end of the talk, and you'll see a chat box down in the left. So if you have a question, type it in. We'll try to answer as many as we can at the end of the webinar, and certainly we will get back to you by email in the next several days if we couldn't answer all of them. And the webinar also is being recorded, so if you want to go back and watch it or forward it to a friend, you'll have the ability to do that when we post it on our PPMD website. Before we start, I want to quickly thank our many partners that we have worked with to get as far as we have with PPMD's Gene Transfer Initiative, which we launched earlier this year, if you recall. As you know, PPMD, we made a significant investment in, and we strongly support gene therapy as a potential therapy to slow or to stop this disease. We have several programs moving ahead, and we hope that each one individually will contribute to a more thorough understanding of gene therapy. And you can see some of the programs listed on the slide. Where all these programs together, we are intending, they're supposed to help get this therapy over the finish line. So many thanks to all the partners that are listed here, including our advocacy partners, companies, and individuals who contributed to our holiday campaign, because that was the big push. And so now it's my pleasure to introduce our two speakers for today's webinar. We have Professor Melissa Spencer from UCLA and Professor Lee Sweeney from the University of Florida. Professor Melissa Spencer received her BS and PhD degrees from UCLA. She's the Professor of Neurology at the David Geffen School of Medicine and the Neuromuscular Program Director in the Department of Neurology at UCLA, and is also a co-director for the Center for Duchenne Muscular Dystrophy at UCLA. Her lab focuses on gaining mechanistic insights into pathogenesis and muscular dystrophies and applying that knowledge towards therapeutic interventions. Her lab has a particular interest in the contribution of the immune system to muscular dystrophy pathogenesis and fibrosis. Through these studies, the lab identified osteopontin as an immunomodulatory protein, promoting fibrosis and inhibiting regeneration. Dr. Spencer serves in many roles that serve the community. She currently serves on the Scientific Advisory Committee for PPMD, Coalition to Cure Calpain 3 and MDA, and she was awarded a Presidential Early Career Award for Scientists and Engineers in 2001. Dr. Lee Sweeney is the Thomas H. Marin Endowed Professor of Pharmacology and Therapeutics at the University of Florida and Director of the University of Florida Myology Institute. Dr. Sweeney earned a master's degree in physiology and a doctorate in physiology and biophysics from Harvard University. He served as chairman of the Department of Physiology at the University of Pennsylvania School of Medicine from 1999 to 2013. Much of Dr. Sweeney's research program is translational in focus. and has produced highly cited research on inherited forms of cardiovascular disease and on the skeletal and cardiac aspects of muscular dystrophy. He has been director of a Paul Wellstone Muscular Dystrophy Cooperative Center since 2005, first at Penn and currently at University of Florida. Dr. Sweeney is actively working on development of therapeutics for DMD. So with that introduction and goals listed for today, I'd like to turn it over to Dr. Spencer, who will start us off.
Okay. So this slide represents some of the main therapeutic targets that are being investigated for drug development to slow disease progression in Duchenne. Some of these targets address the primary cause of disease, which would be dystrophin replacement, while others address secondary causes that hope to slow disease progression. And although gene replacement is expected to make a dramatic difference in slowing disease progression, it may not be 100% efficient, and so some of these add-on therapies may be needed or desirable in combination to have a more powerful effect. So today we're going to focus specifically on gene therapy where either a dystrophin gene, a new healthy gene, is being transferred into a human cell using a viral vector. And we're also going to touch upon CRISPR, which is a platform that can achieve gene editing in live human cells. So before I get into the therapies, I first need to give you a short biology lesson so you can understand why we have had to make some decisions along the way based on knowledge of dystrophin and muscle. So what I'm showing you here is a cutaway of skeletal muscle, which is highly organized into bundles of contractile proteins. So those are the proteins that allow muscle to generate force. And because muscle is a force-generating tissue, it has to be able to bear the loads that are generated. And it's very important that every time muscle contracts, it can sustain those loads. And that is why nature designed a very specialized system to help protect muscle from falling apart every time it contracts. And so encircling each of these muscle fibers is a membrane, which is enlarged down here. And this membrane is peppered with many different proteins, including a complex called the dystrophin-associated glycoprotein complex, or DGC. When you have dystrophin mutations, such as when you have what's represented by this X, you have no protein produced. So when no protein is produced, no dystrophin protein, you lose this whole complex of proteins from the membrane. Now, the way dystrophin anchors those proteins and the way dystrophin helps protect the membrane from damage is by tethering it like a rope. So you can see at this end of dystrophin, it's holding these proteins here in the membrane. That's the C-terminus, and you'll hear this referred to later. And down at this end of the protein, it's anchored to basically the bones of the cell, which we refer to as the actin cytoskeleton. So the most important parts of the dystrophin protein are the ends, what we call the business ends. And you'll hear more about this later in the webinar. We also know that you can move certain parts of the central region, and it's still a fairly functional protein, and we will talk about this later on. So dystrophin helps to give the muscle cell some structural integrity to allow it to sustain the forces that are generated on it. So a muscle cell that has dystrophin will be strong, and a muscle cell without dystrophin will start to sustain damage. And that is what happens in Duchenne muscular dystrophy. So a little more biology. As you know, DMD is a genetic disease, and different types of mutations lead to disease in our genes. So what I'm showing you here is 46 different chromosomes. The DMD gene, so the causal gene for Duchenne muscular dystrophy, is found here on the X chromosome. And so our chromosomes contain lots of different genes. So chromosome 1, for example, doesn't just have one gene. It has an array of many different genes. Same with the X chromosome. Now, when our genes are going to be expressed into a protein, so these genes encode all the proteins of our body, it has to be unwound. So if you look here, if a chromosome is unwound, you can see that it exposes the letters that make up the code. And each gene has a start and a stop. And that tells the body where the beginning and the end of the gene are. Now, our chromosomes are found in our nucleus. So they're found there and they can't move. And so to be able to express a protein when the chromosome is unwound, it has to be made into an RNA. And that's like a xerox copy of the gene. So the RNA is made from the start to the stop. It's a molecule that can leave the nucleus and go out and be made into protein in specialized sites of the cell. They're like little protein-making factories. Okay, so just a small review. So this represents the chromosome. The gene is found in one site of the chromosome. That's the code. That codes for the dystrophin protein. And that code has to be made into a xerox copy. That's the dystrophin mRNA. And then that, can be, these are all stages of production of dystrophin protein that can be targeted for therapy. So one way to target or address the mutation in dystrophin is to replace it or repair it. And that's what I'm going to be focused on today. We also could trick the cell to skip around the mutation, and that's at the level of the mRNA. And that's, for example, exon skipping therapies act here, which you may have heard of. And lastly, the cell can also be tricked at the process of making the mRNA into protein at these protein-making factories to ignore the mutation. And that's the basis for the new PTC drug that's coming up for its AdCom. All right, but we're going to focus today on this, replacing or repairing the gene. Oops, I'm sorry. So that is what we call gene therapy. It's replacing replacing the gene, adding exogenous DNA into a cell, or it is repairing the DNA that is already there. So how do we get the DNA? We can clone a gene. How do we get it back in the cell? Well, nature has a very specialized way that it can get DNA into cells, and it's through a pathogen or a virus. So viruses, this is what they do. They go into cells, and they use the host protein-making factories to generate viral proteins. So we can capitalize on this and make the virus so it is not pathogenic anymore, so it doesn't make you sick. So you take out all of the viral genes, and you replace the viral genes with the gene that you want to be made. And the virus is modified, it goes in, it does not integrate into the host genome. And that's a very important thing, because if it integrates, it could disrupt other genes. It stays what we call episomal. So it's a very stable bit of DNA that sits around and is made into RNA and then protein. So we need to get the virus carrying the gene into all the cells of the body. How do we do this? Well, one virus that is very effective is AAV, and it's also very efficient in making protein. And it can go all through the body systemically, so that means through the blood system, and it can get to all the tissues. But we don't want it to be made in all the tissues. We only want it to be made in muscle and heart. And so what we do is scientists engineer a specialized green light that allows it to be made in some tissues but not others. So we put this, it's called a promoter. We put a muscle-specific promoter onto the virus, and therefore it will only be made in muscle and heart and not in kidney, lung, etc. There's one other issue that's been a stumbling block that's now been overcome for this AAV, that is the primary virus that is used for gene therapy these days. And that is that AAV cannot carry any amount of cargo or any amount of DNA. It is limited in how much it can carry. And so dystrophin is a very large gene, making a pretty large protein. But we got lucky in a way that this is a structural protein, and we can, based on a lot of work done by Jeff Chamberlain over many, many years, he methodically went through this dystrophin protein and figured out what are the most critical and the least critical parts of the molecule. And he then engineered it so that we could have a very small dystrophin that doesn't have all of the regions of the molecule. He deleted several parts that weren't the most critical. But now he could fit it into the AAV vector. So it went from about 14 kb to about 4.5 kb. And Dr. Sweeney will touch on some more detail of this later on. But one thing I do want to point out is you can see in the central part, it looks like a lot of the same type of domain. We call those repeats. So we know with dystrophin we can get rid of several of these repeats, and it's still fairly functional. And as long as we retain these business ends of the molecule, the most critical region, it's a fairly functional dystrophin. So we now know that we can remove parts of dystrophin, the dystrophin gene, we can reduce it in size, we can put it in an AAV vector and deliver it to the body. So just to summarize this part, when we're talking about micro or mini dystrophin gene therapy, we're talking about replacing the mutated DMD gene with a healthy gene using a virus to deliver it. It stays episomal. It does not integrate. We know that the DMD gene needs to be reduced in size to fit into AAV, and there are several upcoming trials that are going to use different versions that Dr. Sweeney will talk about. And it's taken about 20 years of development to get to this point, but we hope that we think trials will be underway in 2018. Okay, so the next thing I want to talk about is CRISPR-Cas9. This is a powerful new technology that allows scientists to make precise edits in DNA in live cells. And it's based on a bacterial system that was exploited by scientists only about 5 years ago to allow for editing of human DNA in live cells. And that's a phenomenal technological development. And this editing system actually creates a permanent change in the DNA. So it can either cut out a piece of the DNA and replace it with a new piece, or the system can cut the DNA and seal it up without any replacement. And we know that this cutting and replacing with a new piece of DNA, which essentially would take a mutated gene and completely fix it, that's not efficient at all. And right now it's not at all a reality. But we know because of the way dystrophin functions and because of the structure of this molecule, we know we can cut out parts, and they're not so critical for its function. And so the second way is the way that most scientists are now thinking about as the way forward for Duchenne. So let me describe the CRISPR-Cas9 system to you. It's a two-component system, and I'm going to refer to these two components as flags and scissors. So the flags are what we call guide RNA. They're little pieces of RNA that are able to go to certain parts in the genome, and we design them in such a way to go where we want them to go. So they mark precise regions of the genome. In our case, we're trying to mark the DMD gene. And those flags help guide the scissors, which is known as Cas9, and that's a DNA cutter. It's a protein that cuts DNA. And it follows those flags wherever they go and cuts, just blindly cuts wherever the flags exist. So in the case of, as I mentioned, we would cut out this region between the two flags, and then these two pieces would come together and seal up. So we would lose this intervening region. Okay, so let me talk to you a little bit more about the biology again, because I want to explain to you how CRISPR-Cas9 can be used on the DMD gene. So many of you are familiar with this exon diagram because of the exon skipping therapy that has been recently FDA approved. So what I'm showing you here is the 79 exons of the DMD gene. And these are pieces of the gene that fit together like puzzle pieces. Now, there are intervening regions that you don't need to worry about. But the reason that they're cut apart like this is because those intervening regions do eventually get snipped out. So it's very important for a gene to be made into a protein that all of these puzzle pieces fit together in this way. Now, if you lose a piece of the puzzle, such as when you have a out-of-frame mutation, and most deletions cause out-of-frame mutations, you lose the ability of the puzzle pieces to now fit together. So that is when we say out-of-frame, that is exactly what we mean. We mean the pieces don't fit together anymore, and there's no protein produced. You can reframe, meaning you can make the puzzle pieces fit together again if you delete an exon that allows like pieces to fit. So in this case, I'm showing deletion of exon 51, just like Ediplursin. This is what Ediplursin does. So that 49 is a straight piece, and now that fits with another straight piece. So this is how exon skipping therapy works. That acts on the RNA. In the case of CRISPR-Cas9, we're now acting on the DNA. So that's those chromosomes that are in the nucleus. So the CRISPR system goes into the nucleus. It can cut wherever the scientist targets the cuts to be made, and pieces can come together. So I'm giving you the example of exon 51, but in CRISPR therapy, or I shouldn't even call it a therapy yet because we're not even close. In the use of CRISPR, you can cut out as much or as little as you want. It's just a matter of where you design those guides. And so in the end, you go from having an out-of-frame piece of protein, which is not, there's no protein produced here, so this would be the mRNA, out-of-frame, to a protein that is now slightly truncated in the middle, wherever you've made your cut, but it's now back in frame. And in the case of DMD, we would do this in the central rod region here and maintain these business ends. And there are different types of deletions that you will hear about from Dr. Sweeney that can be more favorable or less favorable for the function of this protein and its ability to retain these proteins at the membrane and act as a shock absorber. So that's the other function of dystrophin. So in the future, how do we put this CRISPR-Cas9 in the body? Well, one thing we know is it needs to be made safer than it is currently being used in practice. So scientists use CRISPR in the lab. They use it as a tool. There have been studies done in animal models where it's been put in and it acts on the dystrophin gene and it makes new dystrophin. But the way it's been done so far in preclinical studies is not going to be applied to humans, and we know it needs to be safer. We can add this platform as DNA in a virus, but it can also be added as RNA or RNA protein. And RNA or RNA protein is probably going to be safer than giving it in a virus as DNA. And as I said before, you can give it as a virus, but also scientists are working on non-viral methods that can be used to deliver these different components. And a key aspect of the longevity of this therapy is to target the satellite cells. And we know that AAV does not target satellite cells very efficiently, if at all. And so we really need to focus on getting this therapy to satellite cells, and that is currently not a reality. Another way it can be used is by editing cells outside the body. So you could take out muscle stem cells, edit them, and put them back in. And scientists are working on ways to make this more efficient. It's still in very preclinical stages, but that is one way that CRISPR-Cas9 can be administered. So just to summarize the CRISPR portion, CRISPR-Cas9 is a gene editing platform that can permanently remove one or more exons. It can be applied to a large number of mutations. but as I said, it is still very preclinical, early stages. Multiple strategies can be used to deliver this, and those are all being tested. This would be one of the furthest from trials as far as gene therapies are concerned. And one of the issues is the delivery of the platform and also potential off-target effects. And we know that viral delivery will be very, very challenging because not only do you have the issues of immunity to the vector and the new dystrophin gene that's being given, but also you're putting in a bacterial protein that the body sees as foreign. So it's a very new technology and safety needs to be established, but it's an extremely exciting therapy that we're very hopeful for the future.
Okay, I would like to take over from Melissa, and I want to basically point out that the reason we're here talking about AAV gene therapy with microdystrophin being a reality within the next year really is it has been set up by some recent AAV gene therapy successes. The first successes that you probably all are familiar with is the work of Jane Bennett at Penn, my old institution, where she was trying to correct hereditary forms of blindness. And the reason that this was probably the perfect place to start with AAV is because when you inject into the eye, there is no immune response. The eye is an immune-privileged space, and so the immune system never saw the AAV, but it bore out the fact that the AAV was capable of expressing new genes stably in these cells. Kathy High's work, also at Penn and at CHOP, was focused on hemophilia. It took a bit longer to get it working than Gene's work because of the immune response. In fact, Kathy's initial hemophilia trials delivering factor 9 did not work because as she escalated the dose of virus, there was an immune response, and those infected cells in the liver that received the virus were destroyed by the immune system. She later realized that she could make it work by applying immune suppression before she gave the virus and keeping it there for a little time afterwards, and that was enough to keep the immune system away while the virus was visible to the immune system. And because of that, she has now been successful in her hemophilia treatment. But the key trial for this community, the one that really is pivotal in allowing the regulators to even entertain a systemic AAV gene therapy trial in Duchenne, is Jerry Mendel's type 1 SMA trial. This is a trial where in infants, Dr. Mendel gave a very large dose of AAV, enough that it would infect essentially every cell in the body. And this would never, I think, have been allowed by the regulators in Duchenne if not for the fact that Jerry has seen no adverse events. He's used mild immune suppression, and that has been with steroids, high-dose steroids, again, transiently, just a little bit before you give the virus and keep it for a while after you give the virus, and then you can withdraw it. And that has allowed the delivery to be what appears to be very efficacious. He's not yet published his results, but the regulators have access to the results, and because of the safety, they're now going to allow us to go forward with high-dose systemic AAV in DMD patients. So I think Dr. Mendel has to be commended for basically opening the door for this community by first moving to type 1 SMA. So let's talk a little bit more about the micro-dystrophin gene therapy. First of all, as Melissa said, we're trying to replace dystrophin and deliver it with a virus, but we can't use all of dystrophin, so we have these so-called microdystrophin constructs. It's not dependent on the type of patient mutation, like with the exon skipping or with the nonsense suppression. It'll work in any patient. Now, the caveat, as some of you know, because of the nationwide trial has some exclusion criteria around certain types of mutations, it's not because it couldn't work, but they're trying to be ultra-conservative. And I'll come back to this in a little bit about why they're trying to exclude some patients, even though it potentially could work in those patients. And one of the big reasons, is the next point, is that prior exposure to AAV may prevent the therapy from being administered. So that is to say, if in the real world you've been infected by AAV, then your body has seen it, it's made antibodies against it, and those antibodies, if they're circulating, will stop you from having a reinfection. So in this case, even though the antibodies were made against wild, AAV that was not engineered, it will still stop the engineered AAV from infecting. And so we have to get around that. But if you start with young children, the likelihood of them having seen AAV before is low. This type of therapy is not going to be permanent. If you know much about muscle, you know that the more you use muscles, the more you have to renew them, the more you have to repair them, the more the satellite cells have to come in and build new muscle. And so when you do that, you lose the nuclei and the muscle that was there before. And so over time, you may lose the nuclei that receive the AAV DNA. Now, it's going to be totally dependent on the usage of the muscle, and from animal studies, we think it could at least be a decade or maybe more before you would lose it. But we really don't know how long it's going to last in humans. So we're thinking that eventually you're going to have to re-administer the therapy. As Melissa's already mentioned, the clinical trials, and several of them are likely to start in 2018. So what is the difference between micro and mini-dystrophin? Well, this is really historical, this terminology. Those of us who are old enough like myself who have started in the gene therapy world when adenovirus was our only vector before AAV came along, we had made what were so-called mini-dystrophins. They were smaller pieces than the full-length dystrophin, because adenovirus couldn't fit the full-length dystrophin, but it could fit a much bigger piece of dystrophin than can AAV. And so when adenovirus failed for a variety of reasons as a gene therapy vector, and we moved to AAV, we had to make the dystrophin construct even smaller. And so to distinguish them from the ones that had been used in adenovirus, they were called microdystrophins rather than mini-dystrophins. We think that the more the better, that the bigger the piece of the dystrophin, the better. But in fact, the microdystrophin work in animals would have you believe that they are sufficient to give you a lot of the benefit of even the full-length dystrophin. Not all of it, but a lot of it. And so we think it's going to be able to stabilize the muscle to a very great extent. There potentially are advantages of the microdystrophin over an exon skip product, and as I'll go into later, because some of the products from exon skipping, although they're bigger, they're not as stable as the microdystrophin. And furthermore, with the microdystrophin gene therapy, you're going to be able to express very high amounts of the dystrophin. So that means it'll diffuse broadly throughout the muscle cell, which means you might not have to infect all of the nuclei to get dystrophin throughout the muscle cell. There are two main factors in determining the functionality of these small truncated dystrophin proteins. One is we think you really have to retain the two ends, as Melissa already mentioned, the part that binds to the axon cytoskeleton at the beginning or at the end terminus, and the piece of the C terminus, not the whole C terminus, but the piece of the end that binds to the membrane-spanning protein complex, the dystrophin glycoprotein complex. And then you have to have some of the middle. The middle is the so-called spring, and it's shown here as these little blue discs, but what they are is they're molecular springs that allow the protein to extend under force and not break the membrane, but basically extend the protein. So it's like having a big spring and shock absorber all in one. It can transmit the force, but as the force gets too high, then the springs sort of come apart rather than tearing the membrane apart. So obviously the more you have of them, probably the higher loads you can withstand. But nonetheless, having a small number of them is far better than having nothing. And so it's going to be quite protective just to have one of these microdystrophins with either 4 or 5 of these springs instead of the normal number of springs. And that's about all we can get is 4 or 5 of these spring-like regions into the microdystrophin that fits into AAV. So what are the limitations here? Well, as I said, bigger could be better, but it's not always better. Even though these microdystrophins are quite small compared to some of the exon skip products, they've been designed to be extremely stable. And so stability is really the key. You want the protein to be there and to stay there. The reason Becker muscular dystrophy patients can have a wide range of disease severity is precisely because of this. They have essentially deleted some of the exons, but they're still in frame, just like we're trying to create with exon skipping. And yet some of them have disease that's indistinguishable from Duchenne, and others have disease which almost is not noticeable at all. Some of them have been diagnosed very late in life. And so this huge gamut that you see in phenotypes is really related to the stability of the protein. Now, this is what the protein sort of structure looks like for these repeats or these springs. So I'm not going to explain this in great detail, but just I've colored alternating repeats blue and red here, and then the hinge regions, which are these flexible regions in green. And this is just showing you the front end of the dystrophin rod or repeat. And what you can see is that these amino acids are lined up in these 3 cylinders. That's because these repeats form a protein structure that is known as a triple helical repeat. So the individual amino acids make these helical regions that then stack upon each other. And then the reason this creates a spring is because if you start to pull on these things, then the forces that hold them together will withstand the pull and then slowly give as you increase the force. The problem with exon skipping is that the exons do not code for individual repeats. They code for partial repeats. And so if you delete 1 exon or 2 exons, you don't delete an entire repeat, you delete part of a repeat. That leads to an instability, and depending on what you delete, it can be more or less of a problem. If you unfold the repeat, for instance, it can unfold its neighboring repeats, and then you get this large area that's permanently unfolded and is a target for proteolysis. That's why many of these Becker-like proteins don't accumulate very much in the cell, because they're chewed up by the proteolysis machinery. Others just
Lee, what's proteolysis? Can you just tell everybody what proteolysis is?
Oh, yeah, the muscle, any cell will break down protein that appears to be defective or unfolded. And so the fact that you create a protein that's unfolded from the start means that the protein degradation machinery is going to attack it and get rid of a lot of it. And that's why you just can't, in some of these Becker-like constructs, accumulate very much protein, because even though you're making it, the cell is constantly chewing it up. So the ones that fold better are going to be more stable, and some of them do fold very well. We know that because some Becker patients are either mild or almost non-presenting, and others are very severe. So we know that exon skipping is going to create a range of protein stability. We don't have a very good way to predict it. If you look at the exon chart again, now I've shown where the repeats are versus where the exons are. You see what the problem is. None of the exons really correspond with repeat boundaries. I mean, here's one that sort of does. If you took out exon 36 and 7, you pretty much would cleanly take out repeat 13. And so that would be like no disease at all. Whereas other repeats, if you take them out, other exons, if you take them out, you really would destabilize. Like here, if you took out only 47, you would totally destabilize repeat 18, which might then in turn destabilize 17 and 19. So that's the problem, and that's why it's hard to predict how good a protein you're going to get out of exon skipping. And in fact, if we're going to do this with CRISPR, we really need to make animal models where we skip the exons that we intend to skip in patients and try to understand which ones are going to make really great proteins and which ones are going to make bad proteins. Now, going back to the potential for microdystrophins, these are some of the microdystrophins that have been designed and tested in animals, and likely you'll see these in some of the trials. Zhao Zhao, who is one of the founders of Bamboo Therapeutics, has done a considerable amount of work with this particular microdystrophin that has 5 repeats and these 2 hinges still intact. Nationwide is going to use a construct that was designed by Jeff Chamberlain that he's done extensive amounts of work with. It has 4 repeats and 2 hinges. And then Solid is using a more recently designed construct by Jeff Chamberlain that incorporates 5 repeats and also a region that is thought to be important for blood flow regulation in the muscle to see if that can be restored to normal as well. So these are the microdystrophins that are likely to be entering these trials. Some of these have been confirmed and some of these haven't. But in all cases, there's going to be a muscle-specific promoter that expresses, as Melissa said, in the skeletal muscle and in the heart, followed by the dystrophin mini gene, followed by components that allow the RNA to be processed and stable and then translated into protein. Now, some of the limitations, and these are limitations whether we're talking about gene therapy for microdystrophin or for CRISPR if you're using AAV, is the fact that AAV itself will cause an immune response against what's called the capsid. That's the outside of the virus. That's what the body sees first, is the protein that surrounds the virus. As I said earlier, the trials, especially the SMA trial, has shown that that if you use immune suppression prior to getting the virus, you can get in. But so far, we haven't been able to figure out how to then come back and re-administer. We think it's possible, and people are working on technologies to do it, but it may require that you use a different type of AAV that the body won't recognize when you come back. The other problem we have is that if you have prior exposure to AAV in the world, then this could prevent the therapy from being administered, because if you have high levels of antibodies, those so-called neutralizing antibodies will prevent the virus from ever getting into the muscle cells. We think in the general population that maybe about 20% of people have pre-existing immunity to these AAVs that we're using. That number is going to go down as the boys are younger, because really as you get older, your chances of being exposed to the virus are higher and higher. So the younger the boys, probably the lower the probability that they have pre-existing immunity to AAV. But that'll only come out as we really take a close look at these patients. So there's also the possibility of an immune response to the transgene. So if you're doing microdystrophin, that means that if there's a region in that microdystrophin that was deleted in the patient or never expressed in the patient, then it's possible the body could see that as a foreign transgene and mount an immune response. This is not a definite thing, and even if it did happen, it's not clear that that would destroy the muscle and lose the benefit. But there is some concern about it, at least amongst some of the community, and because of that, I'll talk about at least one of the trials in the early stages is going to try to avoid the possibility. But as I say, this really hasn't been explored very carefully, and it may not be a problem at all. As Melissa said, what is certainly a problem if CRISPR-Cas9 gene therapy were to move forward as it has in mice, is Cas9 being a bacterial protein is going to provoke a massive immune response if you continuously express it. So one thing that can be done, and there's lots of different strategies you can think of, is you need to inactivate the Cas9, make it go away as soon as it excises the DNA. And there are ways that you could possibly do that, but they need to be worked out and tested in animals before you can even think about going into humans. So this is just, let me I know there have been a lot of questions about the exclusion criteria that have been announced for the nationwide trial. Based on the microdystrophin construct that's being used, the initial trials that are going to be conducted with at nationwide are going to accept only patients that have deletions or mutations between exon 8 and 58. Now, the reason for that is, as I said, because of the particular microdystrophin they're using, it's encoded by exons 1 through 17 and by exons 59 and on. And therefore, anyone who has a deletion in this region has no chance of seeing it as a foreign protein, whereas there is a small chance that people who have mutations earlier or later would see it as a foreign protein. It's not a definite, but Dr. Mendel is being ultra-conservative in his first trial because he doesn't want to have any problems that he has any control over, because we don't know what the problems are going to be, so we don't want to create a problem if we can help it. He's also restricting the expression to only the heart and the skeletal muscle using a modified version of the creatine kinase promoter that Dr. Hauska at the University of Washington has created. And I think all of the trials have probably used various forms of muscle-specific promoters, especially to not have the protein expressed in antigen-presenting cells to, again, minimize the risk of an immune response. Now, another question.
Can I ask, before you leave, can I just ask one thing, please, Abby? So, but the intent, like the way we're designing the trials up front, is just to minimize those chances. But if it's an approved therapy, the intent is that we would have overcome any of those issues, and hopefully all will be eligible. Is that what the intent is?
Yes, of course. I mean, the idea is that every patient should be eligible for this therapy. The question is, do you need to do more than Dr. Mendel is doing in the first trial if there's any potential for an immune response, or is that really not even an issue? And, you know, that's a question he probably will answer in his second trial, or others will answer in their first trial. But at least in his initial trial, he wants to avoid asking the question because he's got such a small amount of patients he's going to do. He wants them to be homogeneous in terms of the possible problem. So, but the therapy will be applicable to all patients, and I'm sure that the problem that the microdystrophin itself causes is one that we can overcome. I think the bigger problem is getting the re-administration of virus to work, and that's doable too. And fortunately, I think we'll have at least another decade or so to try to figure it out after these initial patients are treated. So will CRISPR-Cas be permanent, or will you need re-administration just like you do for microdystrophin? Well, it depends is the answer. And that's because there are two targets for CRISPR-Cas. One are the muscle fibers themselves, and then, as Melissa said, the second is the satellite cells. If you want this to be permanent, you have to modify the satellite cells. Unfortunately, right now, with the AAVs that we're using, satellite cell infection is very low efficiency. So if you were to do CRISPR-Cas tomorrow with AAVs, it would not be permanent. You would have the same caveats that you do with the microdystrophin. And in fact, it might be worse than microdystrophin because some of the proteins will be unstable, as I said, and therefore they may disappear before the microdystrophins, and you need re-administration even earlier. Another question that we've been getting is whether or not you can participate in other clinical trials if you participate in gene therapy. Well, the answer is no. I mean, it's basically the same answer as if you've been in any of these other investigational trials. Some of them, you know, with a small molecule, if you wait long enough, it'll wash out. But with gene therapy, hopefully it's not going to wash out, at least not in any reasonable amount of time. And therefore, you wouldn't be able to participate in other trials. But of course, you'd be able to receive any approved therapy that would target anything else that you think is going to be beneficial added on to the gene therapy. And furthermore, if you're excluded from a gene therapy trial, it doesn't exclude you from other trials or other approved treatments. So you might be excluded for reasons that would be of no interest to a different trial. So that's not an issue. What about the impact of age? Why dose younger? Why do what Dr. Mendel is going to do? Well, for one thing, the muscles are more intact. And there is a practical issue besides the fact that there's more muscle to rescue, that as the muscle gets more diseased, you have more and more fibrosis. And what we've learned in animals is that fibrosis is a barrier to the virus delivery. So as there's more fibrosis, you need more virus to get into the muscle. So again, it's not that it's insurmountable, but it makes it harder. Also, in younger children, the likelihood of immune response to the virus is lower. As I said, the longer you've been on Earth and the longer you've been exposed to the real world, the greater the likelihood that you've seen AAV in the real world, and therefore you'll have an immune response to it. Now, a possible downside, and this is, I think, even a bigger downside for CRISPR-Cas, unless you hit the satellite cells, is that the muscles are rapidly growing in these young children, and that will dilute out the number of nuclei. If you think about it, the nuclei you hit are the ones that are in the muscle the day you give the virus. If those nuclei aren't there yet because the muscle hasn't grown, then you can't hit them. But the good thing about the microdystrophin is that you're going to be making very high amounts of it, and it's small enough that it's going to diffuse probably broadly within the muscle cell. So even if you only have 10% of the nuclei that are infected, it may be enough to cover the whole muscle. With exon skipping with CRISPR-Cas, you may need a larger percentage of nuclei than that because, in fact, you're not making very high amounts. You're making the normal amounts because you've actually modified the normal gene. So otherwise, there's really no difference between using AAV to deliver CRISPR or to deliver microdystrophin. The issues are basically the same. It's all about AAV delivery. It's not about what you're delivering. What about steroids? So as you know, the immune suppression protocol that Dr. Mendel used in the SMA trial very, very effectively, and what he's going to use in his trial, and I imagine others are going to use, is transient immune suppression with high-dose steroids. Some of the boys probably will not be on steroids, and they will just be on them transiently in this case, because you'll start them before the virus and then leave them on the steroids for a while after giving the virus. The idea is to make the outside of the virus, this coat protein, you want them to go away before you take away the immune suppression. If the boys are already on steroids to begin with, they probably won't be on as high a dose, so they'll transiently go on a higher dose, and then they'll come back down afterwards. But of course, the hope here is if the gene therapy is successful, then they can come off of the steroids. They can be weaned off after this initial period of steroids to suppress the immune response to the virus. That's our hope. We don't know if it's going to be that successful, but if the SMA trial is any guide, then it may well be that successful. Can we make enough virus to treat everybody who needs it? And does either CRISPR or microdystrophin use more virus? Well, the simple answer is, if you were talking about today, the answer is no. But if you look at what's going on, if you look at the excitement in the gene therapy space, and if you look at the industry involvement, what you see is that many AAV production facilities are now being built. So our capacity is going to grow every year, and we're going to be able to make more and more virus. And so it won't take very long before we will be able to make enough virus to treat every patient. Obviously, as the boys get bigger, you need more virus, but it doesn't matter whether you're talking about delivering CRISPR or microdystrophin. It just depends on the size of the person that you're treating. You'll need more virus. So just to wrap it up, talking about microdystrophin versus CRISPR as the payload for AAV. In one case, you have a micro-dystrophin. It's a small protein, but designed to be stable. In the other case, where you're doing permanent exon skipping, you make a product that could be very stable or might not be. If it's very stable, then this could be superior to microdystrophin in that it could be a very big, very stable protein. But if it's not very stable, then the microdystrophins expressed at high levels is going to have a big advantage. As far as immune response, you got the potential immune response to the vector. Well, you know we'll get immune response to the vector. It's just a matter of getting around it. And that's the case in both situations. In the case of microdystrophin, you have this hypothetical immune responsiveness to microdystrophin, and it may happen, but we don't know if it's going to prevent the therapy from being effective. It may well not. There are enough patients that have antibodies that have endogenous exon skipping that makes you think maybe it's not a problem. There's a highly likely immune response to Cas9, so its expression is going to have to be inactivated. Safety, immune response is a major safety issue with the microdystrophin. Immune response, as well as off-target effects from Cas9 cutting where it shouldn't cut, is potential with that type of therapy. Persistence, as I said, we would expect at least a decade if you get high levels of gene transfer with the microdystrophin. If you don't modify the satellite cells, then the persistence with CRISPR-Cas is going to depend on how good the dystrophin is. The better the dystrophin is, the longer it will last. If you hit the satellite cells, it potentially could last forever. Retreatment, as I said, eventually will be necessary. Right now with either strategy. If you can figure out a way to hit the satellite cells, it won't be necessary with CRISPR-Cas in the someday future. But right now we have to figure out, and I think we have a decade to figure it out. We have to figure out how to re-administer these viruses to make this an ongoing therapy to be lifelong for these patients. So with that, I'll stop and turn it back over to Abby.
Thank you both, Melissa and Lee. I think that was a really fabulous talk. We do have some questions, and we don't have very much time left, but if everybody can stay on, we'll try to get through as many as we can. So here's a hypothetical. You're in the microdystrophin trial, but then you're going along, I don't know, maybe year 1 or something, and then CRISPR-Cas9 gets going at the trial. What happens with the AAV, especially if both therapies are using the same AAV? What does the patient do? So I think the question is, well, can you switch from trial to trial? But they're both using the same AAV. So what would the patient be able to do in that situation?
Lee,
well,
want to take it?
You wouldn't be able, no one would allow you to switch from one trial to the other. I mean, that would just complicate. So if you do AAV therapy for one indication, you're not going to go into an AAV trial for another indication. Now, if you're in an AAV for one type of therapy and it doesn't work, and another type of AAV therapy does work and is approved, then you obviously would be allowed to then receive that type of AAV therapy, with all the sort of caveats about re-administration having to be overcome.
Okay. So another question. So the CRISPR-Cas9 system, and maybe Melissa, since you went over that first, I'll direct this question to you, but Lee, feel free to chime in. So the Cas9 is the cutting part of it, the enzyme cutting part of it. So we have heard of another part of the CRISPR system called CRISPR CPF1. So is there if you know about the differences between those two scissors, if you will, is one better pinpoint delivery? How are they different, if you know?
So that CPF1 is just a different kind of Cas9, and so whether you might use one or another might depend on your type of deletion. All of this is all very, very theoretical right now. Nobody has that choice, right? So, yeah, it's just a slightly different type, and it's going to cut in a slightly different way.
Okay. Would CRISPR be effective in BMD, in Becker patients?
Is that for me?
Either of you.
Okay. So the way that we're thinking about using it right now, it will depend on the type of BMD. So you have BMD is basically clinically defined, but most patients have in-frame deletion. So if you're going to be applying CRISPR, the way we're thinking about it now is to create an in-frame deletion. And so you could potentially take a bad BMD mutation, so one that creates an unstable dystrophin, so it's in frame but the dystrophin is not stable, and create a more stable dystrophin. That might have a slight effect. In the future, when we're very, very good at this, and I'm thinking a long, long time from now, you could potentially replace the missing regions with CRISPR, but that's for the future, for sure.
Yeah, but what a severe Becker patient could be eligible for is the microdystrophin, because that would correct the severe Becker patient just as it will the Duchenne patient. Because the reason that Becker patient is severe is because, as I said, even though he's in frame, the dystrophin that he's making is very unstable, and so it's not going to compete with the microdystrophin. If you express the microdystrophin, going to be very little of the Becker protein there, and so the microdystrophin will basically be allowed to do the same thing it does in the absence of dystrophin, which is to reconstitute the dystrophin glycoprotein complexes throughout the muscle cell. So definitely a severe Becker is a great candidate for the microdystrophin therapy.
Okay, we are running over a little bit. I don't know how much time we can run over, but there's a question talking about it. I think Melissa, maybe both of you both talked about how CRISPR is a permanent cut to or a change to the DNA. Exon skipping is not. They both have potentially similar outcomes unless you're creating a truncated protein that puts the protein back in frame. Does making the changes to the DNA potentially have more impact or efficacy than making changes to the RNA, such as we do in exon skipping?
Yes. I mean, it would be a permanent change to the DNA, but for that permanent change to be sustained, it means the cell has to be sustained. And for the cell to be sustained, you have to have enough dystrophin to keep it alive. And so if you have, for example, a very inefficient, so let me go back. So muscle is a big, long, cylindrical cell with lots of nuclei and also has these satellite cells, these muscle stem cells. If you target those muscle stem cells, then those cells can divide, make new muscle, and make daughter cells. And so you could potentially have a lifelong supply of corrected stem cells. But if you're correcting the nuclei within those big muscle fibers, and you only correct a few of those fibers and you only make a little bit of dystrophin, your fiber's going to die and you're going to lose those corrected nuclei. And so it really depends on high efficiency, very excellent dystrophin production, meaning the type of dystrophin, the type of deletion you're making. But optimally, you would be targeting the satellite cells to get your long-lasting permanent correction. Was that too technical?
No, I think that was okay. I have another question, a couple questions that come in about treating duplication. So if you look at gene therapy with transgene Micro-Dystrophin versus a CRISPR-Cas9, which is one more applicable?
Hey, Abby, you're breaking up a bit.
Oh,
yeah, totally. I did not. Yeah, what was
I can't really
question?
understand the question.
Is this better?
Yes.
I just want to know about some questions came in about duplications. So how do either of these therapies, or would either of these therapies treat duplications?
Yeah, well, with microdystrophin, it really doesn't matter what the mutation is, be it a duplication, deletion, point mutation in the splice site. It really doesn't matter why you're not making dystrophin. For CRISPR, a duplication does create some issues because you'd like to take out the duplicated region, but not the normal region. But it still might allow you an opportunity, depending on where the duplication was, to take out the duplicated region and the original region and restore the reading frame. So you probably couldn't reconstitute a full-length dystrophin very easily, but you could make a truncated dystrophin, potentially.
You could potentially, if you had intronic regions that you could target that were unique.
Yeah, that's true. If the duplication created a situation where you could re-splice it to get the duplicated copy out, you might be able to. But that would be a case-by-case basis.
Yeah, I was just going to say, that sounds like true personalized medicine.
Yeah, it would be.
Yeah. I think we're probably going to have to close up soon because I don't want to take too much of your time, Dr. Spencer and Sweeney. But Dr. Spencer, here's a question for you. Which exons specifically are considered critical for function? I think maybe that got answered a little bit in the second part of the presentation, but maybe a quick review.
Okay. Well, let me get to the slide we have. I think the key thing is really by domain. So there's these actin binding domains at the N-terminus, and those are on the slide here, I think. Or is the slide showing? Yeah. So see where it says actin binding? You can see it's repeat 4. And there's the cysteine-rich region towards the end of the gene where it says cis-rich. Those are really critical, but it's also the way it's folded. You need a certain number of repeats. You need a certain number of hinges to have a decent molecule. I hope that answers the question.
Okay. And I think we did it for if people who want the answers to that question. I think we, if you go back when we post the webinar and go through it, I think we probably answered, give you some more information on that. But email us if you still want more information. I think that we have to stop now. But we do know that our next webinar, and there will be some communication coming about this, will be with Dr. Jerry Mendel on September 6th, and he'll be talking about his gene therapy trial at Nationwide. So look for some information on that. It's tentative, but we think that's the next one. And I want to thank our speakers very, very, very much for the thoughtful presentations and all the work they put into this. And as I said, we will try to get back to you with all the questions that came in and answers and post them on the website when we post the webinar recording. I hope you enjoyed this webinar, and I look forward to more like this. So thank you very much.
Yeah, well, thank you, Abby. And I must say to everyone still listening that I think the advent of these gene therapy trials is the most exciting thing that's happened in a long time for this disease.
I agree. It's a really exciting time, really hopeful time. I would also suggest you check clinicaltrials.gov if you want to know about exclusion criteria for particular trials once they're up there.
All right, everybody. Thank you very much. Have a great afternoon.
Bye-bye.
Bye-bye.
Bye.